Hello everybody and welcome back to another exciting edition of Ed Puzzle Lecture Notes. Last time we were doing diffusion, and today we're going to do something that's very similar. It's almost exactly the same. It's called facilitated diffusion. And speaking of that, this is our learning objective of the day. Describe the process of facilitated diffusion. Now hopefully you remember, in diffusion, substances moved with the concentration gradient or from high concentration to low concentration. You also should remember that since it's going from high concentration to low concentration, no energy is required. So, for example, if these dots were a solute, you can see there's a lot of dots on the outside of this cell and there's a very few dots on the inside. These dots all by themselves, we'll say it's oxygen, are going to want to move from high concentration out here inside to the low concentration inside the cell. Now, with diffusion, with simple diffusion, it is going directly through the cell membrane. And remember, the cell membrane looks something like this. Remember, it's made out of phospholipids. Remember, the head is hydrophilic. It likes water. And the tails are lipid tails. They're hydrophobic. They do not like water. So in simple diffusion, it's going from high to low, and it's going directly through the phospholipid bilayer. So this right here would be the phospholipid bilayer. So these are things like oxygen, CO2, fats. They can go directly through the phospholipids. Alright, so the good news is that facilitated diffusion is almost the same thing as simple diffusion. They're both a type of passive transport. Remember, in passive transport, no energy is required from the cell. And in passive transport, substances move from high concentration to low concentration. There is only one difference between facilitated diffusion and simple diffusion. That is, these things need help. They need a channel or a carrier protein. They need some sort of door to allow these large particles to cross the cell membrane. So it's exactly the same as simple diffusion. We just need a protein. We need some sort of either a channel protein or a carrier protein to allow these molecules to come in and out of the cell. So here's a common situation where glucose, a sugar, is attempting to cross the cell membrane to enter the cell, but it cannot get through the phospholipid bilayer. And, glucose is asking very nicely, hey, can you let me in, please? And the cell membrane says, no, you're too big. Plus, you're polar. The lipid tails block polar molecules, even water. So glucose cannot get through the phospholipid bilayer. But the cell membrane gives them some good advice. You need to find a transport protein. You need to find a channel or a door in order to let you in. And that's what facilitated diffusion is. Alright, so what exactly is facilitated diffusion? That is when substances move with the concentration gradient. And what does that mean? They're moving from what concentration to what concentration? Let's see if you remember. You are right. Whenever you're going with the concentration gradient, you're moving from high concentration to low concentration. Now let's see if you remember this. Is energy required from the cell whenever substances are moved from high to low concentration? Let's see if you remember. You're right. The answer is no. No energy is required whenever you move with the concentration gradient from high to low concentration. And what is the main substance that crosses the cell membrane this way? Glucose, which is a sugar. it needs a protein because it's both too large and it's also polar. So if you're too large, you cannot get through the phospholipid bilayer, and if you're polar, you also cannot get through the phospholipid bilayer. And in case you're curious, the word facilitate means to help. And the reason why it's called facilitated diffusion is because it is diffusion that needs help from some sort of protein to allow that substance or that molecule to enter. So just a little diagram that shows you. So here's glucose. Eventually you're going to need to know its formula. It's C6H12O6. It is a sugar. And even if it tries, it cannot get through the phospholipid bilayer. It is too large and the lipid tails block it. But luckily for it, it's got these proteins right here. Now right here it's called a channel protein. I don't know about that. But anyways, these glucose molecules can get into the cell through these proteins. All right, I think it's a good time for a brain break. So let's do a little riddle. What gets wetter as it is drying? So what thing gets wetter as it is drying? Let's see if you can figure that out. And the answer, of course, is a towel. All right, so now back to types of transport proteins. So on the previous slide, it said it was a channel protein that allowed glucose here to come in. But I didn't lie, but the picture lied. Those were actually carrier proteins. Now you're going to need to know the difference. Honors, you will. Regular bios, don't worry about it. But carrier proteins work by they allow one glucose molecule to come in at a time. So it's not like all the glucose can just rush through the door all at once. Only one glucose molecule can come in at a time. That's because the protein actually undergoes a shape change. So when the glucose comes in and bonds in there, the protein changes shape and allows one glucose to come in. Now obviously, if only one can come in at a time, these are not very fast. So carrier proteins are kind of slow because they allow only one molecule to come in at a time. But it's still going down the concentration gradient from high to low concentration. Now the other type of protein that allows substances to move in and out of the cell are called protein channels or channel proteins. So remember, on the last one, it was a carrier protein. Carrier proteins are very slow because they only allow one molecule to come in at a time. It's kind of like a single file type of deal. Now, all you need to know here is channel proteins or protein channels allow substances or solutes to enter much more quickly because if they are open, all of the solutes can just rush in. They don't have to wait for that protein to change its shape. They can just all rush in as fast as they can. All right, thank you very much for listening. Hope you enjoyed it, and I'll talk to you next time. And a little review time. So I'm going to ask you a few questions, and hopefully you get them all right.